Hello guys. So today we are going to discuss about amino acid metabolism. We know that nitrogen occurs in foods, tissues and body fluids mostly as proteins. It also occurs in body fluids and urine as non-protein nitrogenous substances that include urea, ammonia, etc. We can classify amino acids as essential amino acids and non-essential amino acids. Essential amino acids are those amino acids which cannot be synthesized in the body, so it should be supplied in diet. It is required for normal growth and development. Non-essential amino acids are those amino acids which which can be synthesized in the body. It is formed in body from fat and carbohydrate breakdown. So essential and non-essential amino acids each contains 10 amino acids each. Essential amino acids are methionine, arginine, tryptophan, threonine, valine, lysine, isoleucine, phenylalanine, histidine, lysine. And non-essential amino acids are alanine, asparagine, aspartate, cysteine, glutamine, glutamate, glycine, proline, serine, and tyrosine. So, among essential amino acids, arginine and histidine are also semi-essential amino acids, as these are synthesized in small amounts in our body. Now, we will be looking on to nitrogen catabolism. Nitrogen catabolism of amino acids consists of the removal of their alpha amino group as ammonia by transamination and deamination processes. Ammonia that is released is converted to excretory products such as urea and uric acid through several pathways. And the carbon skeletons which left behind from these amino acids form gluconeogenic intermediates such as pyruvate, oxaloacetate, etc., or any ketone bodies. Now we will go on to transamination process. Transamination involves the transfer of amino group from an amino acid to a keto acid to generate a new amino acid and a new keto acid. And the enzyme which catalyzes this reaction is the transaminase enzyme. This enzyme requires PLP as a cofactor. If we look on to this, we can see that in the illustration, we can see that the amino group of the amino acid has been transferred to the keto acid, and this first amino acid has been converted to keto acid, and we get a new keto acid of the first amino acid and a new amino acid of the first keto acid. So this is what transamination is. Now we can look at two examples of enzyme transaminases, and first one is GOT or AST, and its full form is glutamate oxaloacetate transaminase, and AST is aspartate transaminase. GOT is also known as AST, and this enzyme catalyzes the transfer of amino group from the amino acid aspartate to alpha ketoglutarate, which is a keto acid, and result in the formation of oxaloacetate and glutamate. So if we see this illustration, we can see that aspartate has been changed to oxaloacetate by receiving a keto group, and alpha ketoglutarate has changed to glutamate by receiving an ammonia group, an amino group. So this is how GOT or AST catalyzes transamination. The second is GPT or ALT. GPT is glutamate pyruvate transaminase and ALT is alanine transaminase. This enzyme catalyzes the transfer of amino group from alanine, which is an amino acid, to alpha ketoglutarate, which is a keto acid, to form pyruvate and glutamate. So we can see here that alanine has changed to pyruvate by accepting a keto group, and alpha ketoglutarate has changed to glutamate by accepting an amino group. So this is how GPT or ALT catalyzes transamination reaction. Now we will go on to deamination reactions. So deamination is the removal of amino group from an amino acid as ammonia. In transamination, only shuffling of amino groups among amino acid and keto acid occurs, but in the case of deamination, liberation of ammonia occurs for urea synthesis. So the liberated ammonia is used for the urea synthesis, and the deamination reaction is of two types: oxidative deamination and non-oxidative deamination. Oxidative deamination is the process where the amino acids are oxidized to their corresponding amino acids at first. Then this amino acid react spontaneously with water to give a keto acid and ammonia. So we could see here that first of all an amino acid is there, then FAD oxidizes this amino acid in the presence of amino acid oxidase enzyme and form their corresponding amino acid. This amino acid reacts with water with the liberation of ammonia spontaneously and forms a keto acid. So here an ammonia molecule is released. So the second one is non-oxidative deamination. Here several groups of enzymes catalyze the deamination of amino acids, and here no oxidation occurs. Here the enzymes itself catalyzes the reaction without any kind of oxidation reactions. If you look on to two examples, first example is aspartate enzyme. It deaminates non-oxidatively and it liberates ammonia from aspartate and forms fumarate. So if we could see here that aspartate in the presence of aspartase enzyme has been converted to fumarate with the liberation of an H3 molecule, that is ammonia molecule. This is one example. Next example is glutaminase enzyme. This enzyme hydrolyzes glutamine to glutamate and ammonia. So we could see here that glutamine amino acid in the presence of glutaminase enzyme and water liberates ammonia and forms glutamate. So these are the two examples of non-oxidative deamination. There are several other examples also which you can refer later. So after these processes we get nitrogenous end products, and the ammonia which is liberated is the result of nitrogen catabolism has been converted to nitrogenous end products, and it can be urea or purine like uric acid according to the species. For example, ureotelic organisms excrete out urea, and uricotelic organisms excrete out uric acid, and ammoniotelic organisms would give out ammonia as their nitrogenous end product. So according to the species, the end products varies. So this is all about a general introduction of amino acid metabolism. So in the next video we will be discussing about urea cycle.